Did you know that some animals reproduce without a partner, while others can change sex at will? The evolution of reproductive function in animals is a fascinating and complex subject that has captivated the attention of biologists and researchers for decades, giving rise to numerous scientific hypotheses. This vital function, which lies at the heart of species survival, has undergone countless transformations over the millennia, shaping the incredible diversity we observe in the animal kingdom. Dear traveler, welcome. Today we're going back to the origins of reproductive function in the earliest animal ancestors. By exploring paleontological and genetic data, we can trace the roots of sexual reproduction, which emerged hundreds of millions of years ago. This evolutionary history, marked by milestones such as the appearance of gonads and secondary sexual characteristics, gives us a better understanding of how reproductive function has evolved over geological eras, thanks to evolutionary factors such as natural selection, genetic drift, and sexual selection. The mechanisms of natural and sexual selection have played an essential role in shaping reproductive behaviors and morphologies. By examining how these forces have affected reproductive traits, we can better understand why some species have evolved asexual modes of reproduction, while others have opted for complex reproductive systems. We'll delve into this evolutionary history, analyze the variation in reproductive strategies, and reveal the mechanisms that have sculpted this extraordinary diversity. But before you set off on your next adventure, remember to like the video and subscribe to the channel so you don't miss a thing. Thank you, and have a great trip! Although we tend to think of living organisms as adults and of reproduction as the creation of a new adult that resembles the previous generation, it's important to note that every living organism is an organism in its own right throughout its entire life cycle, from fertilization of the egg to adulthood, and not just for a brief period of that cycle. Reproduction is not just one stage in an organism's life history, but its entire history. It is essential to note that only the DNA of a cell is capable of replication, and even this replication process depends on the action of specific enzymes, which are themselves derived from DNA. Consequently, the reproduction of all forms of life must be seen in the context of time. What is replicated are series of copies, which, like the individual images in a film, evolve in a precise and orderly fashion over time. A few examples highlight the considerable diversity of life cycles in living organisms. They also demonstrate that various phases of the life cycle can evolve, and that these changes are not limited to the adult stages. One such variation concerns minimum size, i.e., the size differences between gametes and asexual individuals. Invertebrates have a wide variety of life cycles, particularly those that undergo metamorphosis, a radical physical change. Butterflies, for example, go through 3 distinct stages: caterpillar, chrysalis, and adult. A notable feature of this development process is that during the transition from caterpillar to adult, most of the caterpillar's tissues break down and serve as a source of energy for the next phase of development. This starts when certain small structures called imaginal discs in the larva begin to grow into the adult. Thus, the butterfly essentially goes through 2 phases of growth and development: larva and chrysalis adult, as well as 2 smaller phases, fertilized egg and imaginal discs. This phenomenon is similar in sea urchins, where the larva, called the pluteus, has a small varicose bud that develops into the adult, while the pluteus tissue disintegrates. In both examples, the organism can be said to be living 2 life histories, one built on the remains of the other. Another example of the life cycle observed in certain invertebrates highlights the concept that the major distinctions between organisms do not always lie in the physical appearance of the adult, but rather in the variations in its overall life history. Let's take the Obelia coelenterate, a species of cnidarian, as an example. From an egg, it develops into a colonial structure called a hydroid, consisting of a series of branched, hydra-like organisms known as polyps. Some of these polyps specialize to become reproductive polyps and leave the colony as free-swimming jellyfish, carrying eggs and sperm with them. As with caterpillars and sea urchins, the life cycle of the Obelia jellyfish comprises 2 distinct phases: sedentary, branched polyps, and mobile jellyfish. In related coelenterates, the medusa form has completely disappeared, leaving only the polyp stage to ensure direct reproduction by carrying eggs and sperm. In other groups of coelenterates, the polyp stage has been eliminated, and the jellyfish directly spawns other jellyfish, without passing through a sedentary phase. There are also intermediate forms between these extremes. The importance of biological reproduction can be fully explained by natural selection, as set out in the concept of evolution, in particular through the theory of natural selection formulated by Charles Darwin. Darwin realized that for evolution to take place, it is essential that living organisms are able to reproduce, but also that the resulting copies are not all identical, i.e., that they exhibit variation. In this context, the most suitable variants would contribute most significantly to future generations in terms of the number of offspring. In order for this selection to endure through successive generations, Darwin also understood that these variations had to be transmitted from generation to generation, even if he hadn't yet elucidated the exact mechanism of heredity at the time. What's more, the degree of variation is of particular importance. In line with what is known as the compromise principle, itself influenced by natural selection, it is necessary to maintain a balance in the amount of variation. Too little generates no change, while too much obscures the benefits of a specific combination of inherited traits. Of the various mechanisms for controlling variation, all of which involve a delicate balance between different forces, the most effective is sexual reproduction, present in most plants and animals. In the course of evolution, where reproduction and variation are 2 fundamental characteristics not only necessary for natural selection, but also subject to it, sexual reproduction has proved perfectly suited to generating an appropriate amount of variation. It also enables the rapid integration of new combinations of traits within a population. An examination of the evolution of organisms from their single-cell beginnings in ancient times reveals a trend towards multicellularity and increasing size in plants and animals. Once cellular reproduction evolved into multicellular growth, multicellular organisms developed a mode of reproduction that could be more accurately described as reproduction throughout the life cycle. The increase in size brought with it numerous mechanical requirements, necessitating selection in favor of intense efficiency. The result has been a marked increase in the complexity of organisms. In terms of reproduction, this has led to a substantial increase in the possible variations in cellular reproduction during the evolution of the development process. The increase in size also implies an extended life cycle, with a great diversity of models at different stages of this cycle. Each phase of the life cycle is adaptive, with naturally selected characteristics enabling the organism to survive. In some cases, such as caterpillars and butterflies, distinct life cycle phases, separated by metamorphosis, have evolved, and the duration of these phases can be modified by natural selection, as seen in various coelenterate species. The efficiency of reproduction in order to contribute effectively to subsequent generations is another factor that has evolved through natural selection. For example, some organisms may produce many eggs, of which only a small proportion will survive, while others produce very few, or even a single egg, which they take care to protect throughout its development, thus considerably increasing the chances of survival. Both reproductive strategies have their respective advantages and disadvantages. Many other considerations linked to the natural history and structure of the organism, determined by natural selection, dictate which strategy is best suited to a particular species. It's essential that every species finds a balance, producing enough offspring to survive as long as overpopulation and disease don't become a problem. The number of individuals in some populations fluctuates cyclically, but generally remains within upper and lower limits. How natural selection regulates the number of individuals is a matter of great interest, involving various factors that influence the rate of reproduction. Since hereditary variation is mainly encoded by genes located on chromosomes, sexually reproducing organisms require a single cell stage in their life cycle. At this stage, the haploid gametes of each parent can fuse to form the diploid zygote. This process is also often observed in asexually reproducing organisms, but in this case, the asexual reproductive organs, such as spores, are small in size, favoring their efficient dispersal. Diversity of variation is regulated in a variety of ways, all involving a complex set of balanced factors. These factors include the mode of reproduction, whether asexual or sexual, the rate of genetic mutation, the number of chromosomes, the degree of exchange of chromosome segments, known as genetic recombination, the size of the individual, corresponding to the complexity and duration of the generation cycle, the size of the population, the level of inbreeding, and the relative proportions and position of haploidy and diploidy in the life cycle. Clearly, then, the mode of reproduction influences the degree of variation and vice versa. Together, these two elements allow natural selection to take place, and this selection, in turn, modifies the mechanisms of reproduction and variation. The various types of asexual reproduction enable organisms to reproduce quickly and efficiently, without needing a partner for fertilization. However, this also limits the genetic diversity of offspring, which can be a disadvantage in terms of adaptation to environmental change. Asexual reproduction occurs in prokaryotic microorganisms such as bacteria, as well as in some eukaryotic organisms, whether unicellular or multicellular. Animals have developed various methods of asexual reproduction. Binary fission is how prokaryotic cells and some protozoa reproduce. It's a form of asexual reproduction in which a parent cell undergoes division to give rise to two perfectly identical daughter cells, each of which has the potential to grow to the size of the original parent cell. For single-celled organisms, this is the only method used to produce new individuals. Many organisms use this primitive mode of reproduction, a notable example being bacteria. Bacterial fission involves chromosome replication, segregation, and cell division. Protozoa, another group of organisms, also use binary fission for reproduction. In the case of protozoa, the process shares similar fundamental steps with that of bacteria. However, protozoa differ from prokaryotes in the need to duplicate and divide their mitochondria. They also exhibit variations in the way their cells divide. In amoebae, for example, cytokinesis can occur in any plane, demonstrating a type of irregular binary fission. However, in the case of longitudinal fission, the example of Euglena, a common genus of flagellate green algae, is illustrative. Serratium, another protozoan, is distinguished by oblique cytokinesis. Paramecia is an example of a protozoan using a type of transverse binary fission. Budding represents a mode of asexual reproduction in which a new individual is formed from the outgrowth of a part of a cell or a region of the body, leading to a separation of the original organism into two individuals. In some species, the ability to produce buds can extend to almost any part of the body, while in many cases, this ability is restricted to specialized areas. Initially, a protrusion of cytoplasm, or multiplying cells called a bud, takes shape, gradually developing into an organism that is a replica of the parent. This new individual may later detach to live independently, or the buds may remain attached, forming aggregates or colonies. Budding is a phenomenon observed in a few unicellular organisms, such as certain bacteria, yeasts, and protozoa. However, it is interesting to note that several metazoan organisms, including some cnidarian species, regularly adopt budding as a method of reproduction. Fragmentation represents another form of asexual reproduction in which a parent organism divides into fragments, each capable of developing independently into a new organism. These fragments grow and mature to form an entirely new generation, with characteristics identical to those of their parents. This mode of reproduction follows a three-stage process. The process begins with the creation of fragments, followed by their growth, and finally ends with a transformation of the fragments into mature individuals of the species. Fragmentation is a common method of reproduction in many coral colonies, both hard and soft. In addition, many sea anemones use division or fragmentation as a means of reproduction. Other examples of reproduction by animal fragmentation include starfish and planaria. Parthenogenesis most often involves the development of a new individual from an unfertilized gamete, making it a unisexual form of reproduction. It has been observed in almost all major invertebrate groups, including hemochordates, and with the exception of protochordates, and frequently occurs in alternation with bisexual reproduction, which involves the union of gametes. In some species where males are completely absent, reproduction seems to occur exclusively by parthenogenesis. Species that switch from parthenogenic to bisexual reproduction or alternate between these two modes of reproduction, often referred to as heterogeneous species, generally do so in response to variations in population density, food availability, or other environmental conditions. Among the most famous examples of parthenogenesis are rotifers. In some rotifer genera, males are totally absent, while in others they appear briefly and often seasonally in the population. Females predominate, or are the only sex present in the population for most of the year. Since no meiosis division takes place during egg maturation, these eggs are diploid, meaning they contain the full number of chromosomes. They give rise to new diploid individuals without requiring a chromosomal contribution from a male gamete. That is, diploid parthenogenesis. Even in the presence of males, spermatozoa are unable to fertilize the eggs, as the latter are already diploid. In the event of environmental stress, such as seasonal changes, some females produce eggs that undergo reduction division, creating eggs with a haploid number of chromosomes. To give rise to new females, these eggs must be fertilized by a male gamete. Once the new individual is mature, it is likely to reproduce parthenogenetically. However, in the absence of males in the population, haploid eggs can develop into haploid males, which then participate in bisexual reproduction. Haploid parthenogenesis. Eggs produced by bisexual reproduction are often referred to as winter eggs because of their thick shells, which protect the embryo in adverse environmental conditions. Summer eggs resulting from parthenogenesis have a thinner shell. As a result, bisexual reproduction takes place only occasionally to ensure the survival of the species. Nematodes, especially free-living species such as some dioecious soil nematodes, exhibit a type of parthenogenesis known as gynogenesis. In this mode of reproduction, the spermatozoa produced by the males do not fuse with a female's haploid egg, but simply activate it to initiate development. The result is the birth of haploid females. Parthenogenesis, which seems rare in annelids and mollusks, is more common in arthropods. Cladocerans, such as water fleas, have a reproductive cycle similar to that of rotifers. As long as environmental conditions remain optimal and food is abundant, females produce other females by diploid parthenogenesis. When conditions become unfavorable, males begin to appear in the population, triggering bisexual reproduction. The precise trigger for the appearance of males is not yet fully understood. Fertilized eggs, protected by a highly resistant shell, enter a dormant phase and can survive severe environmental conditions, including extreme temperatures and desiccation. Improved environmental conditions lead to the emergence of new females that reproduce by parthenogenesis. The ability to form a resting stage regulates population density. Whenever the food supply becomes insufficient due to an overpopulation of parthenogenetic females, bisexual reproduction is triggered, followed by a dormant phase. Insects offer numerous examples of parthenogenesis of varying complexity. One of the most fascinating is observed in bees. Unfertilized eggs develop into males called drones, while fertilized eggs give rise to workers, maintained in a state of non-reproduction by the secretions of the queen bee, the reproductive female. Life cycles involving transitions between parthenogenesis and bisexual reproduction are also observed in many homopteran and dipteran species, such as flies. Aphids, which are homopterans, have a seasonal cycle comprising a bisexual winter phase and a parthenogenetic summer phase. Some species carry out each phase on a different host plant, and the onset of these phases is largely influenced by factors such as temperature variations, day length, and food availability. In the midge, a variety of flies, the bisexual phase takes place in adulthood, while parthenogenesis occurs in the larvae, a phenomenon known as pedogenesis. Adult females lay fertilized eggs, from which larvae hatch, their ovaries developing, while the rest of the body remains a larval form. The ovaries of the larvae release eggs that enter the larval hemocoel, where they develop while feeding on larval tissue. When they reach a sufficient stage of development, the young resulting from parthenogenesis emerge, either as larvae that continue their parthenogenetic reproduction, producing similar larvae, or as male or female larvae that mature into adults and reproduce bisexually. Sexual reproduction is a mode of reproduction in which two organisms, usually of different sexes, contribute genetically to the formation of offspring. This involves the fusion of special reproductive cells, called gametes, which contain half the genetic material of each parent. Male and female gametes, such as spermatozoa in males and eggs in females, combine to form a new individual. Sexual reproduction results in great genetic variability among the offspring, as it mixes the genes of both parents. This variability is essential for the adaptation of species to environmental change, as it increases the chances of offspring survival by introducing new genetic combinations. As such, it offers advantages in terms of evolution and genetic diversity, but generally requires a greater investment of time and energy than asexual reproduction due to the formation of gametes, the search for a partner, and fertilization. Gamete union is an essential process in sexual reproduction in animals, allowing sperm and egg cells to fuse together. forming a new individual. However, the mechanisms that facilitate this union have evolved in different ways in different animal species. This diversification of gamete union mechanisms is the result of organisms adapting to the pressures of natural selection. Natural selection favors individuals with efficient reproductive mechanisms, enabling them to maximize their chances of reproduction and transmission of their genes to the next generation. The mechanisms of sperm transfer and fertilization are well studied in only a few sponge species. In these species, sperm are produced by the male reproductive organs, then expelled from the body into the water via a complex network of canals. When a sperm enters another sponge, or the one from which it originated, it is captured by a special flagellated cell called a choanocyte. The choanocyte then completely envelops the sperm, loses its flagellum structures, and moves to the internal tissues where the egg has matured. There, the choanocyte containing the sperm fuses with the egg, achieving fertilization. In freshwater coelenterate, sperm are also released into the water and transported by currents to other individuals. However, unlike the sponge mechanism, coelenterate eggs develop within the epidermis or superficial tissues and are exposed to nearby sperm in the water. Many species of marine coelenterates release both sperm and eggs into the water, where fertilization takes place. However, some jellyfish offer some protection to the eggs. After expulsion from the reproductive organs, the eggs are temporarily lodged in the epidermis beneath the organism, where fertilization and the first stages of development take place. In all flatworms, fertilization takes place internally. In species without a female duct, sperm is injected and fertilization takes place inside the tissue. However, most flatworms have a sophisticated system of male and female ducts. Typically, the male duct passes through a penis-like organ, and sperm is transferred during copulation. In the case of parasitic species, which often have difficulty finding a partner, self-fertilization is used as a means of reproduction, also known as hermaphroditism. Sperm and egg fuse in the oviduct, which then secretes a yellow substance around the zygote. Male nematodes generally have a pair of copulatory structures called spicules, which guide the sperm during copulation. Some male nematodes also have a lateral expansion at the posterior end, called a copulatory bursa, which surrounds the female during copulation. Other males wrap their tail around the female at her genital opening during copulation. Unlike many other aschelminths, nematodes have amoeboid spermatozoa, meaning that the contents of their cells appear to be in constant motion. In some annelid species, the reproduction of polychaetes, marine worms, is synchronized with lunar phases. During the reproductive period, the body of both sexes undergoes differentiation into two distinct parts, an anterior part and a posterior part called the epitoke, which houses the reproductive organs. When the moon reaches a particular phase, the epitoke detaches from the rest of the body and swims to the water's surface. It seems that female epitokes induce male epitokes to release sperm, triggering the expulsion of eggs. Fertilization takes place outside the bodies of the individuals and is so well orchestrated that numerous epitokes emerge at the water's surface virtually simultaneously. When they reach sexual maturity, oligochaetes develop a special organ called a clitellum, which is characterized by a saddle-shaped glandular thickening located near the genital orifices. During mating, the clitellum secretes a mucus whose role is to keep the worms together during the sperm exchange. After mating, the clitellum continues to play an important role. It secretes a substance that forms a cocoon around the worm in which the eggs and sperm are deposited. The worm then manipulates this cocoon until it slides over its head. The ends of the cocoon seal, allowing fertilization and development to take place inside. When it comes to gamete fusion in mollusks, several methods are used. Marine bivalves, for example, simultaneously release sperm and eggs into the marine environment, while some freshwater clams appear to be capable of self-fertilization. However, among the marine gastropods of the Scalidae family, an unusual reproductive variety stands out. They produce two types of spermatozoa. A large spermatozoon with a degenerated nucleus serves as a vehicle for transporting numerous small, fertilizing spermatozoa through the water to the oviduct of another individual. Male cephalopods, on the other hand, have specially modified arms for transferring spermatophores. In squid, for example, the right or left fourth arm is adapted to this function. After an often elaborate courtship display, the male squid uses this modified appendage to extract spermatophores from their reserve in his own body and deposit them in the female's mantle cavity. A cementing substance released by the spermatophore fixes it firmly to the female's body, close to the oviduct. In some species, the male may even sacrifice his arm in the process, and the female may manipulate the eggs with her arms. In the case of land snails, unusual behaviors have evolved in parallel with sperm transfer. Prior to copulation, calcium carbonate darts are forcefully propelled from the genital opening of each mating individual to lodge in the partner's viscera. Although snails adopt a mating position, sperm transfer can only occur when each snail has been stimulated by the impact of the dart. Like mollusks, arthropods display a wide variety of methods for achieving sperm fertilization of eggs. Although arthropods have relatively few specialized structures for sperm transfer, many species stand out for their high behavioral complexity. For example, male and female scorpions engage in a complex ritual. In some species, the male produces spermatophores, which he anchors to the ground. During the ritual dance, the female is positioned over the spermatophore. The male then exerts pressure on the female until the sperm packet is forced into her genital chamber, where it attaches itself with small hooks. Fertilization thus takes place inside the female's body. In the case of some spiders, males use their pedipalp, an appendage used for gripping and crushing, which contains a bulb and an extensible, coiled structure called an embolus. At the start of mating, the male dips his pedipalp into his own sperm, then inserts the embolus into the female's genital opening, transferring the spermatozoa into her seminal receptacle. The female deposits the collected sperm with her eggs in a silken cocoon, which she attaches to her own body or to an object such as a stone or leaf. In other arthropods, such as copepods, isopods, and many decapods, sperm transfer generally takes place after courtship, using modified appendages, gonopods, or spermatophores. For example, copepods grasp the female with their antennae while placing a spermatophore at the opening of the seminal receptacle. For some decapods, fertilization occurs when the eggs are released into the water. In insects, fertilization is always an internal process, but there are many variations in the way sperm is delivered to the female. In some species, males produce spermatophores, which are deposited in the female's copulatory bursa or vagina. The wall of the spermatophore ruptures, allowing the sperm to swim to the seminal receptacle. In other species, sperm are introduced directly into the seminal receptacle via an intromittent organ. In still other cases, the spermatozoa, without spermatophores, are deposited in the copulatory bursa and then migrate to the seminal receptacle. In all cases, the sperm are retained in the seminal receptacle until after fertilization. An exception to this sperm transfer pathway occurs in insects, which inject sperm into the female's homocele, i.e., the space between the body's organs. The sperm then migrate to the ovary and oviduct, where they fuse with the eggs before the shell is formed. Invertebrates have developed a variety of strategies to protect their fertilized eggs in developing embryos, and to ensure a supply of nutrients for young individuals. This adaptation is particularly marked in freshwater and terrestrial species. For example, sponges and coelenterates facing seasonal drought conditions create a resistant envelope for their eggs, preventing water loss. Many turbellarians encase their eggs in a capsule, which they attach to a hard surface where the embryos develop until they hatch. Other turbellarians keep the encapsulated eggs inside their own bodies until development is complete and the young emerge. Parasitic flatworms, as well as most nematodes and rotifers, enclose their eggs in a protective capsule, where development takes place after the parental body has emerged. However, some nematode species are oviviparous, meaning that eggs hatch inside the maternal body. In many species, the amount of yolk contained in the egg and the structure of the egg capsule are linked to the annual seasons. In general, summer eggs have less yolk and thinner capsules than winter eggs. This seasonal correlation is also found in many crustaceans. Freshwater and terrestrial annelids create a protective cocoon for their offspring, often deposited in a humid environment. However, some leeches do not form a cocoon. Instead, they attach the eggs, wrapped in a protective membrane, to the underside of the parent. During development, the adult leech undulates its body to allow water currents to flow over the young, which can be used for aeration. Freshwater mollusks have the option of surrounding their eggs with a protective envelope or incubating them directly. Bivalves release their mature eggs into their gill chambers, where they are fertilized and embryonic development is completed, thus ensuring protection. Cephalopods such as squid and octopus attach their eggs to a surface, then generate regular jets of water to keep the egg masses free of debris and potentially oxygenated. Some echinoderms also incubate their eggs until the young hatch. Arthropods have a wide variety of strategies for ensuring the survival of their offspring. These include brood pouches, commonly found in brachiopods, isopods, and amphipods. These pockets can be a part of the carapace or form by enlarged plates on the sternum. Crayfish, on the other hand, attach fertilized eggs to their fins and transport them as they are incubated by the female. The most elaborate adaptations for embryos are found in terrestrial arthropods, notably insects. While some species simply lay their eggs and abandon them, others keep the eggs encapsulated inside their bodies during early development. Some species are viviparous, meaning they carry developing young. For example, the eggs of some scorpion species contain little or no yolk, the embryo being fed by the parent in a similar way to mammalian reproduction. In scorpions, part of the oviduct is transformed into a uterus to house the embryo, while another part, close to the female's intestine, absorbs the nutrients passed on to the developing young. Similar adaptations are found in some viviparous insects, with larvae fed by the glandular secretions of the uterine mucosa. The reproductive organs of vertebrates include the gonads and associated ducts and glands. In addition, some vertebrates, including some very primitive fish species, have specific organs for sperm transfer or egg laying. The gonads are responsible for producing the gametes and hormones essential for reproduction. The ducts and associated glands store and transport the gametes, while secreting the substances required for the reproductive process. In addition to these structures, most vertebrates, whether male or female, have a cloaca. This is a cavity that serves as a common final chamber for the digestive, urinary, and reproductive systems, enabling evacuation to the outside world. In the case of lampreys and most ray-finned fish, the cloaca may be small or absent, and the digestive tract has a separate external opening, called the anus. In some teleost species, the digestive, genital, and urinary tracts open independently of each other. Hagfish, closely related to lampreys, have a small cloaca. In many non-mammalian vertebrates, such as reptiles and birds, the upper part of the cloaca is divided by folds into the urogenital chamber and a feeding chamber, both of which open into a common terminal chamber. In monotremes, such as the platypus and echidna, the embryonic cloaca is completely divided into a urogenital sinus, responsible for transporting urine and gonad products, and an alimentary tract, both of which open out independently. The gonads are formed from longitudinal thickenings of coelomic, epithelium, and underlying mesenchyme. Initially, these gonadal crests emerge into the coelomic cavity and are continuous with the embryonic kidney. The germinal epithelium covering these gonadal crests gives rise to the primary sexual cords, which penetrate the underlying mesenchymal tissue. The secondary sex cords develop inwards, just below the germinal epithelium, to form a cortical region. If the gonad is destined to become a testis, only the medullary component differentiates. However, if the gonad is destined to become an ovary, only the cortical component undergoes differentiation. The sizes of adult reproductive organs vary according to the development of certain parts of the reproductive system. In some groups of animals, such as lampreys, hagfish, sharks, rays, and bony fish, differentiation of reproductive parts is extensive, resulting in reproductive organs extending over a large part of the body. In amphibians, reptiles, birds, and mammals, on the other hand, the front part of the reproductive organs at the end of the body generally does not develop, except in toads, where only the rear part undergoes this differentiation process. Some vertebrate species have a singularity in the number of gonads, with a single gonad either located in the midline or on one side of the body. This feature is most common in females. For example, adult cyclostomes, whether male or female, have a single gonad. In lampreys, this is located in the middle of the body, while in hagfish, it is on the right side. In birds, most females have a single gonad. However, male birds generally have a pair of testes. There are exceptions to this rule, notably among falcons, since more than half of mature individuals have two well-developed ovaries. It should be noted that in all bird species, a small percentage of females probably have two ovaries, with reports indicating the presence of this characteristic in owls, parrots, sparrows, and doves. For example, it is estimated that 5 to 25% of doves have two ovaries. Some teleost and viviparous elasmobranch species also have a single gonad. In amniotes, i.e. reptiles, birds, and mammals, the presence of unpaired gonads is unusual. However, some lizards have a single testis, and some female crocodiles have a single ovary. Among mammals, the platypus generally has a single ovary on the left, while some bat species have only a right ovary. There are two possible explanations for the presence of unpaired gonads. On the one hand, paired embryonic gonadal crests may fuse to give rise to a median gonad, as is the case in lampreys and perch. On the other hand, a single gonadal crest can accommodate primordial germ cells by migration, i.e., sperm or immature eggs, with the result that the opposite gonad does not develop, as is the case in chickens and ducks. It has been observed that falcons generally possess an equal number of primordial germ cells in both gonadal crests, resulting in the presence of two ovaries. In lower vertebrates, mature gonads sometimes produce both sperm and eggs, giving rise to what is known as hermaphroditism. This characteristic is more common in cyclostomes and teleosts than in other fish. Teleosts may function as males at the start of their reproductive cycle, then transform into females later on. In some teleost families, sperm and eggs mature simultaneously, but develop in separate parts of the gonad. These fish usually act as males during one breeding season and as females the next. Cyclostomes are generally ambisexual during their juvenile phase, meaning that immature male and female sex cells coexist side by side, or, in the case of hagfish, the anterior part of the immature gonad may be an ovary, while the caudal part is a testis. It is assumed that cyclostomes generally become unisexual at maturity. In amphibians, hermaphroditism is uncommon, although anomalies can sometimes cause it. In vertebrates above amphibians, true hermaphroditism appears to be rare or non-existent. Male and female duct systems may sometimes be non-existent. In cyclostomes, some elasmobranch species and teleosts such as salmon, trout, and eels, gametes are transported to the posterior part of the coelomic cavity, often by means of cilia, tiny hair-like structures, and are expelled via two funnel-shaped genital tracts located near the base of the tail. In anurans and amniotes such as reptiles, birds, and mammals, and even in some teleosts, the testes are mainly composed of seminiferous tubules. These tubules are coiled, and their walls contain cells responsible for sperm production. The walls of the tubules in the testes are made up of several layers of a type of cell called germinal epithelium. In these tubules are the cells responsible for sperm production. There are also Sertoli cells, which play a nutritive role, helping the developing spermatozoa. The seminiferous tubules, which start at the outer layer of the testicles, curve inwards. They join to form a network of collecting tubules called the testicular network. In some amniotes, such as the rat, the seminiferous tubules may be open and travel a zigzag path from the network to the periphery and vice versa. On average, these tubules are about 30 centimeters long and rarely have interconnections between them. In many mammals, the tubules are grouped into lobules separated by walls of connective tissue. This organization enables a large quantity of germinal epithelium to be concentrated in a restricted space. In immature males and in adult males outside the breeding season, the seminiferous tubules are barely visible and the epithelium is inactive. However, in some species, spermatogenesis, i.e. the production of spermatozoa, can take place continuously throughout the year. An active epithelium may display all stages of sperm development. Within the lumen of the tubules are the tails of numerous spermatozoa, whose heads are embedded in Sertoli cells, free spermatozoa, as well as fluid that is probably resorbed. In certain groups of animals, such as cyclostomes, most fish and amphibians with tails, the organization of male reproductive cells is different from that of the vertebrates previously mentioned. Instead of seminiferous tubules, there are a large number of small sacs, or cysts, in which sperm cells develop. Unlike other vertebrates, the cells lining these cysts are not germinal in nature, i.e., they are not directly involved in sperm production. Instead, the cysts serve as protective environments for sperm development. In the process of sperm production, germ cells move from a permanent germinal zone, which may be located at the periphery of the testes, or along a ridge at the tip of the testis, depending on the species. Once inside the cysts, these germ cells begin to multiply significantly, producing large numbers of sperm. This multiplication leads to pronounced swelling of the cysts, making them appear whitish. As a result, the whole testicle takes on a swollen, granular appearance. When the spermatozoa reach maturity, they detach from the walls of the cysts in which they have grown and swim freely in the fluid contained within these sacs. Eventually, these small sacs rupture, releasing the sperm into ducts. In some fish, such as cyclostomes and teleosts, sperm are ejected into a special cavity called the coelomic cavity. Once expelled, the empty cysts can either regenerate to form new sacs or be populated by new germ cells. Leydig cells, also known as interstitial cells, are present in most, if not all, vertebrates. They are thought to be a major source of androgens, or male hormones. However, Leydig cells are not always easily discernible, and in some bird species, they can only be identified by electron microscopy. In the testis of the rat, and probably in many other animals, there is a network of small blood vessels called capillaries. These blood vessels are arranged so that the blood that is supplied the Leydig cells, located in the testicle, moves to the tubules, which are the important parts responsible for sperm production. It is therefore entirely possible that the hormones produced by these Leydig cells have a direct effect on the reproductive cells, influencing the sperm production process. With the exception of mammals, the testes of vertebrates are located inside the body. This arrangement is also observed in many, sometimes all members of mammalian orders, such as monotremes, insectivores, hyracoids, edentates, sirenians, cetaceans, and proboscideans. Some male mammals, such as most marsupials, ungulates, and primates after infancy, have a special structure called the scrotum, where the testes permanently reside. However, some mammals have a pouch that allows the testes to descend and retract using muscles. This includes some rodents, such as ground squirrels, most if not all bats, and some primitive primates, such as lorises and potos. Rabbits, most rodents, and some insectivores do not have scrotal sacs, but do have a structure called the inguinal canal. This canal allows the testicles of these animals to be pulled or retracted into the abdomen in the event of injury or other dangerous situations. When the testicles are pulled up into the inguinal canal, this causes temporary swelling of the area between the anus and the perineum. However, it's important to note that a small number of mammals keep their testes permanently in the perineal region, instead of pulling them up and down in the inguinal canal. The scrotum is an essential part of the body for regulating testicular temperature. It cools the testicles by bringing the warm blood flowing towards them closer to the blood vessels through which the cold blood leaves the testicles. This temperature regulation is important, as sperm cells develop optimally at temperatures slightly lower than normal body temperature. It's essential to note that when both testes fail to descend into the scrotal sacs, a condition known as cryptorchidism, this can lead to permanent infertility. In other words, the affected animal cannot produce sperm effectively. Birds, like mammals, are homeothermic animals, which means they maintain a constant body temperature. Their testes are located close to the air sacs, which are extensions of the respiratory tract. This enables birds to regulate the temperature of their testes in a similar way to the scrotum in mammals, adjusting blood flow to maintain optimal conditions for sperm production. Reproduction in the animal kingdom illustrates a fascinating and complex story of adaptation and innovation in the face of changing environmental challenges. Over billions of years, living organisms have evolved from simple modes of reproduction, such as asexual cell division, to much more complex systems of sexual reproduction, offering increased genetic variability and improved survival strategies. The diversity of reproductive methods is reflected in the incredible variety of approaches to carrying and rearing offspring, from egg-laying in nests to internal gestation and live births. This complex evolution testifies to the ability of living organisms to adapt and respond to environmental challenges, while highlighting the delicate interactions and trade-offs inherent in life on Earth. As we look to the future and continue our exploration of the natural world, we're ready to discover even more wonders and mysteries. Reproduction in animals is a never-ending story of remarkable discoveries, surprises, and adaptations, reflecting the ingenuity of nature and the richness of life on our planet. In the next part, we take a deeper dive into the mechanisms and curiosities of animal reproduction, revealing even more astonishing aspects of this fascinating facet of biology. We'll take a look at the reproductive strategies of vertebrates. How do these often more familiar but equally astonishing creatures ensure their descendants? What evolutionary innovations have shaped their reproductive methods? See you in the next episode.